The overall aim of the following experiment is to give adherent properties to bacteria in order to facilitate their separation from liquid media. This is achieved by first designing a synthetic Curli Operon to bypass complex genetic regulation and enable easy and massive production of sticky polymers. As a second step, adherent bacteria are visualized and quantified on polystyrene, which allows for a quick estimation of the efficiency of the synthetic device. Next, adherent bacteria are visualized on glass by confocal microscopy in order to directly observe the biofilm and to gain information on its structure. Results are obtained that show a significant increase of adherence in the strain harboring the synthetic device, based on both adherence percentage measurement and microscopic observations.
The main advantage of the synthetic biology approach over existing methods like conventional cloning is that a construct harboring only the absolutely required genes with appropriated signals can be very quickly obtained.
This method can help answer key questions in the bioremediation field, such as addressing the challenge of removing metal-loaded bacteria from radioactive waste. Generally, individuals new to this method will struggle because basic knowledge about genetic regulatory signals and DNA databases handling are required.
Visual demonstration will focus on the quantification and visualizations of biofilm because these two steps are very difficult to learn because of the fragility of biofilms.
The first step to designing the synthetic